Okay, good morning. So I think I just posted actually a study guide for carbohydrate metabolism session, right? So I know somebody asked whether the textbook is useful or not, but I think you guys want to just focusing on the class notes, right? The content that we discussed in the class. So if you can understand all the material we talk about in class, it should be fine, right? With quizzes and exam. And textbook, I think when you think you need to understand more or more comprehensive understanding of the materials, and I think you can read textbook. And also we have assigned reading, right, in the last slide of each class note. So just follow that list. So what kind of information that you need to read from the textbook, right? And next lecture, right, Thursday, we have a quiz. So quiz basically is just several very straightforward general questions, right? And also the coverage, the quizzes, I think also listed in the syllabus, right? Quiz 1, basically lecture last week's, and today will be testing the Thursday, and similar like for the following quizzes. Always the date, the quiz date, the lecture on quiz day, usually we will not include in the quiz, right? Any questions? Yes.
What is the format of the quizzes? Is it like short response or?
Yeah, the format will be just a short answer, right? Very general questions, straightforward. Okay. Now, so last time I think we stopped here, right? We discussed the two different types of response to the increase the blood glucose levels after meal. First tier response, do you guys still remember? What is the first tier response? Which tissues are involved? First tier response? Sugar, sorry, say that again.
The tissues?
What's
the tissues that are involved? Or?
Yeah, your tissues involved. Pancreas. Right, pancreas is involved, and what happened during the first tier response? After meal? Right, high glucose. And what pancreas does? To release insulin, right? So in response to high blood glucose, and pancreas, the beta cell particularly, synthesize and release insulin to raise the insulin level in the blood, right? So both glucose and insulin levels are increased after meal. So this will consider the first tier response. And a second tier response, which tissues are involved? Muscle and adipose tissue, right? And what these two tissues do in terms of regulation of blood glucose levels? To uptake glucose, right? And this which glucose transporter involved in the second tier response in these two tissues? Group 4, right? And transport glucose in response to insulin, right? And transport glucose from blood into the tissues. And these two tissues particularly important because they are respond to insulin, right? And transport a lot of glucose into the particularly muscle, right? Because we have a large mass of a muscle. So muscle plays a more important role in terms of reducing the blood glucose level, right? So uptake a lot of glucose into muscle and also adipose tissue. So we finished this slide, I think. All right. Okay, so now why group 4 in both skeletal muscle and adipose tissue are insulin dependent? That means only when you have a high blood insulin levels and this group 4 can be activated and transport glucose. So here's the reason, because people, actually scientists, discover that group 4 in the basal state, basal condition, that means when insulin levels are low in the circulation, and the group 4 in muscle cells or in adipose cells usually not stay on the cell surface, but inside of cells, right? So again, when insulin level is low, then group 4, majority of group 4 stay inside of cells, right? So this, let's see. So this represented the cells, right? And this, are the group 4 molecules or vesicles. And under the basal condition, when insulin levels are low, the most of glucose will stay inside of cells, right? Glucose transport group 4 stay inside of cells. And when insulin levels increase during, the fat condition, right, after meal, when we have a high blood glucose level and we stimulate the glucose, stimulates insulin release, so insulin level is high, right, after meal, and insulin can stimulate group 4 translocate from inside of cell to the cell surface, right? So once group 4 present on the cell surface, then group 4 is able to transport the glucose, right? This small dots represent the glucose molecules, and transport the glucose from the circulation or outside cell into the cells, right? So this sort of the mechanism why group 4 is insulin dependent, because insulin, again, without high levels of insulin and most of group 4 transport molecules stay inside of cells, and once insulin level is high, insulin can stimulate the movement of group 4 molecules from inside of cells to the cell surface and transport glucose. And during the fasting, you can see in the cells usually have first of all have a lower levels of group 4, have a less group 4 molecules being synthesized during the fasting condition, right? And of course, you also have less glucose, group 4 on the cell surface for transport glucose. So during the fasting, because of a low level of insulin, the synthesis of group 4 molecules is kind of decreased, is low, and you have less group 4 molecules, right, being available for transport glucose. So again, insulin actually does two things. One is to increase the synthesis of group 4 molecules, and second thing that insulin does is to increase the translocation of group 4, right, to the cell surface. Now I wanted to tell you the other important thing is, let me see, did I, or is it still that? Okay. So the group 4, group 4 translocation to the cell surface, right, you just described, is mostly regulated by insulin, right? In the particularly in the fat cell and also muscle cells. And in addition to insulin, actually exercise is also play a very important role regulate translocation group 4 from inside of the cell to the cell surface in the skeletal muscle. So under the different physiological conditions, so in the fat condition, insulin plays a major role. It regulates group 4 translocation to the cell surface in both muscle and adipose tissue. But during exercise, group 4 can also be moved to the cell surface. But under this condition, exercise condition, it's not an insulin, right? Because insulin level is not that high during the exercise. So insulin is not the major regulator of the group 4 movement to the cell surface in the skeletal muscle, but some other mechanism we're going to talk about later. So muscle exercise, muscle contraction can also stimulate group 4 translocation to the cell surface to uptake a lot of glucose, right? Yeah, question.
What if your insulin's high and you're exercising? Is it max out somewhere?
I think that's a good question. So basically, when you're thinking about the exercise usually muscle utilize a lot of glucose, right, for the energy. So under the exercise, actually blood glucose level is relatively low. So normally, under low glucose level, insulin will not be able to secrete a lot, right? So in general, during the exercise, because of a low blood glucose level, insulin level usually will low. So that would not happen, like you described, unless some kind of, pathologic or disease condition. Yeah, that's good thought, I think. Yeah, question.
So does exercise just stimulate the movement of group 4 in your skeletal muscle, or both your skeletal muscle and your adipose tissue?
Yeah, good question. Only skeletal muscle and doesn't do anything with the group 4 movement in adipose tissue, right? So that's why, for type 2 diabetic patients, we always, encourage them to do the exercise, right? Because type 2 diabetic patient is caused by so-called insulin resistance. Have you heard about insulin resistance, right? That means, you have an normal insulin level, even high insulin level, but the action somehow is reduced in the type 2 diabetic patient. So insulin can function well. So, and because exercise, can stimulate group 4 translocation groups uptake independent of insulin, right? So even your insulin system doesn't work, but if you can stimulate, doing exercise, you still can stimulate group 4 translocation and glucose uptake into the muscle, right? So exercise will work for the type 2 diabetic patient to reduce the blood glucose level. Okay, so different roles of group 2 and group 4 in the glucose homeostasis we discussed already, right? Group 2 basically is expressed in beta cells and also liver, and group 4 mainly in the skeletal muscle and adipose tissue, but these two transporters regulate glucose differently, at different stage or response. First tier response, basically group 2, right? Transport glucose into beta cell and triggers insulin secretion, and group 4 in the muscle and adipose tissue stimulate group, translocation is stimulated by insulin and regulates the transport of glucose and reduce the blood glucose level. So these two transporters work together, right, during fat conditioning after meal and involve this several tissues, pancreas, muscles, adipose tissue, right? Mainly and also liver. Actually, liver also takes up glucose to synthesize glycogen, right, in the fat condition. So all this tissue work together to, to regulate blood glucose level, to not allow the blood glucose level to go too high after meal. And here we have some questions for you to discuss. So what happens to glucose homeostasis if GLUT2, GLUT4 is defective? Let's talk one by one. For example, what happens if GLUT2 level is decreased, and how that patient can manage the blood glucose level, and what about the GLUT4 defective, right? To discuss with your partners and take a few minutes, and we will go back to the answer. All right. You guys have enough time for discussion? All right. Can anybody volunteer to share your ideas of what happened to blood glucose level if the patient, does not have enough GLUT2 expression? Anybody? Glucose will get too high, right? How? Can you explain a little bit? Right. I mean, let's talk about the GLUT2 first. GLUT2, right? But yeah, you're right. You're thinking about GLUT4, right? GLUT2. So which tissues express GLUT2? Which tissues produce the GLUT2? Pancreas, yeah, beta cell and liver, right? So if somehow the patient, the beta cell does not express high enough GLUT2, what will happen to the blood glucose level after meal? It's up, right? But how? What about insulin? Will not be able to produce a lot of insulin, right? Because of lack of GLU2, particularly in the beta cell, so we're not able to transport a lot of glucose into the beta cell and to trigger insulin secretion, right? Yeah.
So on type 1 diabetics, they lack the capability of making GLUT2?
That's a great question. So it's actually not very clear whether the cause of the type 1. The basically type 1 diabetes that the beta cell cannot produce enough insulin, right? But whether they are due to the GLU2 GLU2 expression is not very clear. I don't think in the type 1 diabetic patient their GLU2 level in the beta cell is low, but probably normal still. So yeah, that's great thought. I think a very good question. Okay, so what about the GLU4? I think Teresa, you already addressed, right? GLU4 expression level, which is related to the type 2 diabetes. So type 2 diabetes usually, particularly in the fat cells, have a lower levels of a GLU4 expression, right? So GLU4 and also have less translocation of GLU4. So GLU4 defects can causing the decreased transport of glucose, right, into muscle and fat cells. So then we're not able to reduce the blood glucose after meal. So particularly that happen to the type 2 diabetic patient, right? So when particularly type 2 diabetic patient will have a higher the blood glucose level after meal, or called a postprandial blood glucose level. All right. Okay, so now let's talk about the carbohydrate, particularly here the glucose metabolism in the different tissue. I mentioned a little bit right earlier, the different tissue, they metabolize glucose differently, depend on the metabolic pathway activities or enzyme that involve the metabolic pathway expression level. Some tissue they express high levels of enzyme that involve, for example, gluconeogenesis, right, in the liver and the kidney, but the key enzyme that involved in the gluconeogenesis were not highly expressed in the other tissues. So that's why that only liver and kidney can do the gluconeogenesis, but not the other tissue. So again, different tissue does different glucose metabolism. So let's talk about liver first. So liver is the central organ or metabolic hub, of the glucose, because liver express almost all type of enzymes that regulates the all 5 different metabolic pathways of glucose metabolism, right? So like liver can do the glycolysis, TCA cycle for glucose complete oxidation, and liver can also do the glycogen synthesis pathways, right, and glycogen breakdown pathways, and gluconeogenesis and pentose phosphate pathways, right? So it's very active, and in glucose metabolism can do all 5 glucose metabolic pathways. So can be completely oxidized, right? The glucose once enter the liver, do the glycolysis, convert it to pyruvate, and pyruvate enter this represent the mitochondria. Mitochondria for complete oxidation into TCA cycles, right? So this pathway, but it also be able to do the glycogen synthesis, right, in the fat condition. and also glycogen breakdown and release glucose into circulation under the fasting condition. And the pentose phosphate, right? And also that's the gluconeogenesis that synthesize glucose from precursors like pyruvate and lactate, right? And release glucose during the fasting condition. So you would like you to be able to recognize, the cells, which type of cells, if like I give you some kind of metabolic pathway, and you can recognize this is liver cells or this other type of cells based on, how this cells metabolize glucose. So, and the other things I would think is important, besides metabolic pathways, and metabolite flows is also important. For example, when you see the glucose actually can be released, right, into the circulation, this should be the liver cells, right? Liver will be able to release the, glucose, either from glycogen breakdown or gluconeogenesis, right? And lactate basically can be uptake by liver cells for gluconeogenesis. So you be able to know the flow it should be go out or should be go inside of the cells, depend on the physiological conditions, right, or metabolic pathways. This is the skeletal muscle. If you compare to the liver, it seems like they have a less metabolic pathway, right? But the skeletal muscle is the tissue that can oxidize glucose for energy, and also can store glucose as a glycogen, right? The muscle is also stores a lot of glycogen, and for use as an energy during exercise, right? It kind of purpose is different. The liver stored a glycogen, basically release glucose and for other tissue use as energy during fasting, and muscle stored glycogen will not directly release the glucose into the circulation from the glycogen breakdown, but mostly for the muscle cell uses, right, for energy during exercise. So it's different. And if you look at the metabolic flow, so you only see the influx of glucose, right? So muscle cell basically take up glucose from the circulation and not giving, or release glucose. And lactate, muscle actually release a lot of lactate to the circulation and be used by liver for gluconeogenesis, right? So in the skeletal muscle, lactate usually goes out. And muscle can also synthesize glycogen from glucose, right? All right. I think I asked this question earlier, right? Just wanted to ask again. Does skeletal muscle release glucose into blood? Not, right? And not directly. I mentioned because of the key enzyme that is lacking in the muscle, which is glucose 6-phosphatase, right? We will discuss this again later. But muscle actually can provide gluconeogenic precursors, right? Lactate, right? I just mentioned, and also the amino acids like alanine, and for gluconeogenesis in the liver. So muscle actually not directly release glucose molecules, but it's also contributed to the blood glucose level through the independent, way, right? The indirect way, by releasing lactate and alanine to the circulation and then pick up by liver to make a glucose and then release, right? So in the indirect way and contribute to the blood glucose level. Does that make sense? Right? Okay, brain. So brain in the glucose metabolism, the metabolic pathway is kind of relatively simple. Brain cells can just uptake a glucose and completely oxidize glucose, right, through glycolysis, and TCA cycles complete oxidize CO2 and water. And because brain cell have a lot of mitochondria, right, in the cells. So this seems is the only pathway, that brain cell does for glucose metabolism. Brain cell cannot do glycogen synthesis, cannot do gluconeogenesis, right? So only complete oxidized glucose for energy. And so this is the reason why, brain is heavily reliant on glucose for energy, right? Utilize a lot of glucose for energy. So if we have a lower blood glucose level, hypoglycemia condition, then first organ, we think of the brain, right? We can actually, brain can function well under the low blood glucose level. So that's why we need to maintain the normal blood glucose level to allow the brain, right, have a function normally. Red blood cells is also kind of unique cells, and red blood cells also only be able to utilize glucose as an energy. The reason you probably already noticed, right? Something kind of is missing in the red blood cells. What's missing? Mitochondria, right? So red blood cells do not have mitochondria. So that means red blood cells cannot utilize fatty acids, cannot utilize ketone bodies for energy, because fatty acid oxidation and ketone body oxidation all need mitochondria, right? So red blood cells only utilize glucose for energy, and also red blood cells different from brain cells because of no mitochondria. So red blood cell only does glycolysis, right? Cannot completely oxidize glucose. So the product of the final product of glycolysis in brain cells and red blood cells are different, right? Brain cells, you can oxidize. I can go back to the brain cell. So it can oxidize glucose through the glycolysis to generate pyruvate, and pyruvate can enter the TC cycle and enter the mitochondria, complete oxidized, right? So the product of glycolysis in brain cells is pyruvate, right? And red blood cells, because of no mitochondria. So the pyruvate, generally from glucose, glycolysis, will convert it to lactate, right? So lactate is the final product of red blood cells, right? And lactate, again, once it's generated, so red blood cells also contribute a lot of lactate to the circulation. So lactate and generated and released into the circulation and picked up by liver and for gluconeogenesis, right? Okay, any questions so far? Yeah.
So what did you say? What happens to the lactate? It gets sent to the liver?
Yeah, gets to the liver and for gluconeogenesis, right? Okay, so adipose tissue. Okay, so adipose tissue, we know that it has a group 4, right? And take up glucose. And glucose metabolized in fat cells is also a little bit different, depend on the function, right, of the fat cell. Fat cell, we know, usually store lipids, right? Store the triglyceride. So a lot of triglyceride, are stored in the fat cells. So I think, let me see. Oh, okay. So I am going to actually divide this into 2 slides and talk about it under the fat condition and fasting condition. So it's kind of a little bit different. So let's say in the fat condition, the glucose, right, being uptake into the fat cells and go to the glycolysis pathway, generally pyruvate. And in the meantime, the lots of glucose actually metabolized, generate the glycerol 3-phosphate. So glycerol 3-phosphate is the substrate of the triglyceride synthesis. Remember, triglyceride has 2 parts, right? Glycerol backbone and fatty acids. So this glycerol backbone is actually synthesized from glucose, right, from glycolytic pathway. And pyruvate, once it's generated from glycolysis, can enter the mitochondria and convert it to acetyl-CoA, right, and then enter the TC cycle. And this acetyl-CoA molecules is very important. We will talk a lot later, right? Sort of central metabolites that connect the fatty acids metabolism, amino acid metabolism, and glucose metabolism, right? So like oxidation of glucose, fatty acids, and amino acids can all eventually generate acetyl-CoA. So acetyl-CoA is the building block for fatty acid synthesis, and it's also for the cholesterol synthesis, right? Ketone body synthesis, we talk about later. So under the fat condition, because we have a lot of glucose enter the fat cells, right? So we synthesize a lot of glycerol backbone, 3-phosphate, and some fatty acids. But lots of fatty acids actually is coming from the circulation, right? And then picked up by the fat cells. So fatty acids together with glycerol 3-phosphate and synthesize triglyceride. So under the fat condition, right, the triglycerides is synthesized a lot and stored in the adipose cells. So that will need a lot of glycerol backbone. So glycerol backbone basically is synthesized from gluconeogenic pathway, right? I just wanted to mention that this pathway for fatty acid synthesis is active, but compared to liver, is not that active compared to liver. So liver should be the main tissue that synthesize fatty acids from glucose, right? We mentioned a little bit, I think, right? In the liver, how glucose is metabolized in the liver. In addition to glycogen synthesis, some of the glucose being converted to the fatty acids, right? Lipogenesis in the liver. So there's something happening, during the fat condition in the fat cells. Again, the glycerol 3-phosphate glycerol backbone synthesis is the main pathway, right? Through the glycolysis in the fat condition in the adipose cells. And during the fasting, because we don't have much glucose, right, being uptake into the fat cell because of a lower insulin level, so you have a lower GLUT4 translocation, less GLUT4 on the cell surface, right? So glucose uptake activated kind of role in the fat cell. So, but triglyceride breakdown pathway will be very active during the fasting, right? We wanted to break down triglyceride, release fatty acids. The fatty acids can serve as sort of alternative energy fuel during the fasting for other tissue use as energy. So the glycolysis activity will not be very active in the fasting condition, right? Any questions about adipose tissue? Okay. Let's see. Okay, so now let's talk about the more specific pathways, each pathways of glucose metabolism, right? Talk about the catabolic pathway first. So glucose oxidation, that involves both glycolysis, right, from glucose to pyruvate, and the TC cycle, like complete oxidation, right? So glycolysis, let's talk about glycolysis. So glycolysis is the first step in glucose utilization as energy. So it's also the main pathway for metabolism of fructose, galactose, and other carbohydrates. And the enzymatic reactions can take place in the cytosol of all cell types. So any type of cells in the body be able to pick up glucose and oxidize glucose for energy, right? And the glycolysis occurs mainly in the cytosol, right? And some type of cells depend solely on the glycolysis for energy use. I think we discussed, right? Which type of cells that only, heavily rely on the glucose for energy? Brain, yeah, heavily, and the other type of red blood cells, right? So brain and red blood cells, right? Particularly red blood cells, because no mitochondria, right? They, only rely on the glucose for energy through the glycolysis pathway. So glycolytic pathway involves many biochemical reactions, right? Of course, and many enzymes that involve in these reactions, to regulate these reactions. And what I would like you to remember is that the three rate-limiting reactions in the glycolytic pathway, right? And what is substrate, what is product, and what is enzyme that catalyze this specific called key reactions or rate-limiting reactions, right? Three reactions in the glycolytic pathway, and just for make things simple, that we divided the glycolytic pathway into a two stage, right? So the first stage is called a priming of glucose. So the first stage basically consume ATPs and not produce ATP. Consume ATPs, and during the first stage, the glucose basically has 6 carbons, right? 6 carbons, 6 oxygens, right, and 6 hydrogen. So the 6 carbon glucose molecules is break down into 2-3 carbons intermediates. So this is what happened during the first stage. And there are two rate-limiting reactions, right, during the first stage and catalyzed by two key enzymes. Right. So like this 6 carbon glucose, right, and through the 1, 2, 3, right, 4 reactions, right, and generate the 2 3 carbon intermediates, right, the glyceraldehyde 3-phosphate, and these 2 intermediates actually can interconvert. So basically, it's the 3 generate 2 molecules of a glyceraldehyde 3-phosphate, right, when first stage glycolysis is finished. So the 2 key reactions I marked here. The first is the first reaction, right? The glucose can be phosphorylated to glucose 6-phosphate, and this is the key one of the key reaction and catalyzed by the glucokinase or hexokinase. And these 2 enzymes actually catalyze the same reaction, but just the different tissue. For example, in the liver, glucokinase is more active, right? Level is higher than the hexokinase. So in the liver, glucokinase may be the predominant enzyme that catalyzes this reaction, but hexokinase is more abundant or more higher in the muscle cells. So in the muscle cells, the first reaction usually catalyzed by hexokinase, right? And this reaction actually consumes 1 ATP, right, to finish the reaction. So you need to know the substrate is glucose and product is the glucose 6-phosphate, and enzyme, the hexokinase or glucokinase, and utilize 1 ATP. And the second key reaction is the phosphofructose kinase, right? So phosphofructose catalyzes conversion of fructose 6-phosphate to fructose 1,6 biphosphate. Right? So this reaction also utilizes 1 ATP, and generally this again the 2 3 carbon intermediates, and so again this is the 2 limiting reactions and consume the 2 ATPs during the first stage, right? Let's take 10 minutes break, right? All right. I forgot to mention, actually, I recorded lectures, so I posted. If you guys need to, go through the review, it should be available. And also for Thursday's quiz, I usually will give like 30 minutes, after lecture. Will that be okay? Yeah, give about 30 minutes, after lecture, not like in the beginning of the lecture.
How long does the quiz take?
Say that?
How much time do we have for the quiz?
30 minutes, yeah. Yeah, I think usually you guys, finish very quick, like 15 minutes already. Lots of students faculty done, so. Okay, and that was the first stage, right? And again, 2 key reactions that involved in the first stage of glycolysis, and 2 ATPs are consumed, right, during this first stage, and try to remember actually 2 3 carbon intermediate generated, right? 6 carbon glucose breakdown to the 2 3 carbon intermediates. And then the second stage is actually the producing the stage that produces ATPs, right? And can either produce directly ATP molecules through so-called substrate levels phosphorylation mechanism, or produce the equivalent reducing equivalent like NADH or this kind of molecules, right? During the second stage. So second stage, let's start with this two 3-carbon intermediates, right? And then eventually this 2-carbon intermediates being further metabolized to generate the pyruvate. So I don't think you need to remember each reactions, but again, the key reaction, right? So in the second stage, there is one key reaction that catalyzed by pyruvate kinase to catalyze the conversion of phosphoenolpyruvate to pyruvate, right? So this is the reaction, the key reaction. And two ATP molecules are generated, remember, from one 3-carbon intermediate, right? So one 3-carbon intermediate that produced from first stage can metabolize and generate the two ATPs, right, in these two reactions. So if we're thinking about start from one glucose molecules, and how many ATPs will be generated through this second stage, right? Again, this two ATPs generated from one 3-carbon intermediate. So if we're thinking about a start from the one glucose molecules, so how many ATPs will be generated? 4, yes, right? First stage, one glucose molecules break down to two 3-carbon intermediates, right? And then two 3-carbon intermediate enter the second stage, and each 3-carbon intermediates produce the two ATPs, right? So you have a two actually intermediate, 3-carbon intermediate from glucose. So you should have double, right? Double ATPs. So ATP will, generate four molecules of ATP in the second stage of glycolysis. Does that make sense? Okay. And the other thing is important that NADH, reducing equivalent, is also produced during the first reaction, right? And in the second stage. So NAD will be converted to NADH through the reaction, this reaction. And the final product, whether it's pyruvate or lactate, depends on the cell conditions, right? I think we talked about it earlier. So for example, in so-called aerobic conditions, let's say in the brain cells, right? Because brain cells have mitochondria, so be able to further metabolize the pyruvate. So metabolism occur in the mitochondria, usually considered aerobic metabolism, requires oxygen, right? So again, aerobic metabolism requires both mitochondria and oxygen. So in brain cells, we should consider it's the aerobic condition, right? So in the aerobic condition, pyruvate will be the final product, right? And so, and under the anaerobic conditions, for example, in the red blood cells, because no mitochondria, so aerobic metabolism will not going to happen, right? So then lactate will be the final product. And the muscle, what we're going to talk about very later, exercise session, some type of muscle fibers, and they are actually is high glycolytic because of they have muscle contain less mitochondria. So, but just like if you can remember, that's okay. If not, at this stage, that's fine. Right? But red blood cell is typical anaerobic condition, right? And the other difference is in terms of besides the final product, there are other things actually different. Yes, question.
Are 2 NADH produced from the 2 tree carbon?
Say that again.
Are 2 NADH produced from the 2 tree carbon?
Yes, right. Yeah, thank you. Yes. So, I'm just going to talk about this. So, under the aerobic condition, then let me just ask you a question, see if you understand this, right? So, under the aerobic condition, for example, in the brain cells, how many NADPH generally? Thinking about these cycles, and these cycles only occur under the anaerobic condition. So let's talk about aerobic condition first. So aerobic condition, how many NADH molecules are generated during the glycolysis? See that? No, we have, 2. We have 2, right? 2, 3 carbon intermediates, so 2, yes. So how about under anaerobic condition, how many NADH? No. 0, yes. Thank you. Why 0? Right. Yes. So under anaerobic condition, right, because this reaction is very active, pyruvate is converted lactate, and this conversion requires NADH. So this NADH that generated from this reaction can be used, right, for generation of lactate. So generated here and used up here, right? So basically it's 0. No NADH is generated during the anaerobic conditions, right? Does that make sense? So the total ATP is actually also different, right? If glucose is metabolized in the brain cells versus in the blood cell, right? Any question? Does that make sense? Right? And what about ATPs under both aerobic condition? How many ATPs be? I think we discussed before. 4, right? 4, yeah. ATP always 4, whether it's under aerobic or anaerobic conditions. Well, I'm talking about the directly ATP generated directly from substrate levels phosphorylation. Of course, NADH will eventually also, be used for ATP generation in the mitochondria, right? Okay. Any questions? Okay. Now let's do some calculations. You just take maybe 1 or 2 minutes and discuss to fill in how many ATPs, how many NADH be produced from oxidation of 1 molecule of glucose, and under, of course, in 2 different conditions. Right? One is under the anaerobic condition, and the second slide under the anaerobic condition. Right? Try to finish these 2 slides and discuss with your partners and take a few minutes. All right. So now let's talk about the condition, an aerobic condition, right? So one molecule of glucose is oxidized or through the glycolysis, right? And how many ATPs can be directly generated from glycolysis? Two. Why? Thinking about glycolysis, actually, we divide it like two stage, right? We talk about two stage. And first stage is the stage that consume ATPs, right? So 2 ATPs are consumed during the first stage, and second stage produce 4 ATPs. So net ATP will be, right, 2. So that's why produce 2 ATPs directly from substrate phosphorylation level. Okay, and how many NADH is produced under the aerobic conditions? Two, right? I saw 2 or 3, 4. Two, right? 2. So 2 NADH are produced during the second stage. One from 3 carbon intermediates, right? You have 3 carbon intermediates generated from first stage, so that's why you produce 2 NADH. And do you still remember how many ATPs can be produced in the mitochondria from 1 molecule of NADH? You still remember from, I guess, from biochemistry class? No. We can negotiate. Actually, 2 or 3. Why it's a little bit complicated, right? If NADH is already generated in the mitochondria, the TCA cycle can also produce many NADH molecules, right? And TCA cycle occur in the mitochondria. So NADH generated from TCA cycle can directly go to the electron transport chain, do the oxidative phosphorylation for ATP production, right? So NADH, once it's synthesized already inside of mitochondria, usually generate 3 ATPs, right? Remember this, right? Okay. So, but this NADH generated from glycolysis pathway, which part of cells glycolysis occur? Do you guys remember? Where does cytosol? Yes. So glycolysis occurs in the cytosol, so that means the NADH is also generated in the cytosol, and this NADH cannot, transported into the mitochondria freely. They need some kind of transport system to help NADH that produced in the cytosol and to the mitochondria, and depend on which shuttle system that mitochondria transport NADH from cytosol to the mitochondria. So NADH can either produce 2 or 3 ATPs. We're going to explain this right later. Why sometimes or in some type of cells the NADH from glycolysis only produces 2 ATPs, and other type of cells be able to produce 3 ATPs, depend again on the shuttle system across the mitochondrial membrane. So basically, one NADH molecules produce either 2 or 3 ATPs. Again, depend on the cell type, depend on the transport system across the mitochondrial membrane. So 2 NADH will produce 4 or 6 ATPs, right? So the final ATP molecules it can produce in the aerobic conditions, right? The product will be the pyruvate, right? Is 6 to 8 ATPs, right? Okay, so and the next under the anaerobic conditions, how many ATPs produced from glycolysis? The final product is lactate, right? And under the anaerobic condition, how many ATP can directly produce from the glycolysis? And we just talk about it, right? 2, yes, same. 2 produce 4 in the second stage, but consume 2 in the first stage. So net ATP production would be 2. And what about NADH? How many NADH you would expect? Again, 0, yes, under the anaerobic condition, so 0. So under the anaerobic condition, for example, in red blood cells, 1 molecule of glucose oxidized through glycolytic pathways will only generate 2 ATPs, right? And brain you can generate cells, right? And 6 or 8 ATPs. Okay. Any question? Okay. So I think I'm asking you guys to calculate the numbers of ATPs. I think I just try to help you understand, the metabolic pathway in different cells, and depend on the conditions, and also involve the different organelles like mitochondria involved, and ATP production is also occur in the mitochondria. So okay. Yeah, it's more information about the ATP production. The first 2 reactions, right? The first stage, actually, the negative ATP. You consume ATPs, and these 2 reactions, the second stage, and produces the ATPs, right? So net ATP will be 2. And this table just talk about the directly ATP production, from the substrate level phosphorylation, and not talk about the NADH molecules, right? Okay, so let's see what happens to pyruvate formed from glycolysis. Okay, so I think we talked about this already, right? The pyruvate is a final product of the glycolysis in the aerobic conditions, and this pyruvate will further oxidize. That requires the mitochondria, right? And also requires the oxygen. So pyruvate will be entering the mitochondria and further break down to the acetyl-CoA, right? And acetyl-CoA again is key, right? It's a very important intermediate in the metabolic pathways, so connect the multiple metabolic pathways. And acetyl-CoA then enter the TCA cycles, right? And for complete oxidation, this just represents the mitochondria in the cell. And pyruvate and acetyl-CoA. I think we have a next. Pyruvate, if under the anaerobic condition, right, and pyruvate will be further metabolized to lactate, right, under the aerobic condition. Now let's talk about the other sugars, right? Can also enter the glycolytic pathway, but it's different, right? The pathway is different from the glucose. Here we're going to talk about the fructose. So how fructose is metabolized through the glycolytic pathway, also called a fructocolysis, right? So first of all, I think I mentioned there's two different enzymes, right? Glucokinase, hexokinase. In the liver and skeletal muscle, their abundance is different. So glucokinase is highly expressed in the liver, but lower in the skeletal muscle, and the hexokinase is more abundant in the skeletal muscle. And most importantly, I want to mention as fructokinase. Fructokinase has a similar function as the glucokinase or hexokinase, but utilize a different substrate, right? Glucokinase and hexokinase, the substrate for these two kinases is glucose, right? And fructose kinase substrate for glucokinase is fructose. So fructose basically fructose kinase is metabolized fructose. And fructokinase is very highly expressed in the liver. So the fructose metabolism usually occurs or primarily occurs in the liver. Right? Fructose mostly metabolized in the liver because of the high levels of fructokinase. All right? And if you look at the metabolic pathway, it's also different. So let's focus on this here, the fructose, right? Before talk about fructose, let's just look at the glucose glycolytic pathway. It's just kind of summary, right? Of the glycolytic pathway. So glucose, first of all, is phosphorylated to glucose 6-phosphate by glucokinase or hexokinase, and this is one of the key reactions, remember, right, in the glycolysis. And then glucose 6-phosphate converted to fructose 6-phosphate, and this is the second key reaction, right, in the glycolytic pathway, and so convert fructose 6-phosphate to fructose 1,6-biphosphate, and then to the 3-carbon intermediates, right, by this aldolase A. And fructose is metabolized through a different pathway, right? See, the fructose is phosphorylated by fructose 1-phosphate by fructose kinase. And then fructose 1-phosphate is break down into 3-carbon intermediates, right, by this enzyme, so aldolase B. So you can see that actually fructose metabolized bypasses two key reactions, right, in the glycolytic pathway. So in other words, that fructose is metabolized just through the two reactions, fructokinase and the aldolase hydrolysis. two reactions, then be able to break down the fructose to the 3 carbon intermediates. So fructose metabolism actually bypass this key rate-limiting reactions. So that means key rate-limiting reactions. That means when you have lots of glucose being oxidized to generate intermediates. For example, this fructose 1,6 biphosphate. When fructose 1,6 biphosphate levels increase or concentration increase due to the high levels of glucose metabolism, and this intermediates can inhibit this key enzyme, right? So it can slow down the glycolytic activities, right? So that means our body or cell be able to regulate the glycolytic activities. Once we have too much glucose being oxidized and accumulate the intermediates, then this intermediate can serve sort of a negative feedback mechanism, right, to inhibit the key enzyme to slow down the glycolysis, to slow down the glucose, metabolism. But fructose actually metabolic pathway lacks this kind of a key, enzyme control, right? So fructose can continuously oxidize and produce a lot of intermediates. And this intermediates is, if you still remember, is very important substrate of glycerol 3-phosphate, right? It's the backbone of triglyceride. So fructose metabolites can generate lots of glycerol 3 backbone for triglyceride synthesis. They just lack of control by the key enzyme because we bypass this two key reactions. Does that make sense? Right? So that's why when we consume a lot of fructose, and sometimes some studies showing that the high consumption of fructose can cause the fatty liver disease, can cause the obesity, because your fructose produce a lot of glycerol 3 backbone for triglyceride synthesis, right? It can promote a lot of triglyceride synthesis. So this sort of the bad side of fructose, right? So, and the other thing is, fructose is not like glucose. When we have a high glucose level, and we can stimulate insulin release, right? But fructose is actually does not stimulate insulin release. So fructose is not able to stimulate insulin release. So insulin is actually good thing, when not only push the glucose into the muscle or adipose tissue to reduce blood glucose level, but also push the glycogen synthesis, right? So you push the more glucose to the direction of glycogen synthesis. That in that way you can reduce conversion of glucose to fat. Right? Does that make sense? So so the two things that makes high consumption of glucose to make more fat. One is you bypass the rate limiting reactions, right? So you have lots of 3 carbon intermediates generated, and that intermediates will convert it to glycerol backbone and for triglyceride synthesis. And the second thing is because of the fructose does not stimulate insulin, so will not have a lot of glycogen be synthesized, right? So you have, that again will push more sugar being converted to fat. Yeah, I think I pretty much explained this, right? Okay. There is kind of inherited disease called fructose intolerance that I wanted to mention, since we talk about the fructose metabolism, right? And this kind of genetic deficient disease, the cause of this disease is due to the lack of allelase, right? So allelase here. So allelase we know is to break down, right, the fructose 1-phosphate to the 3-carbon intermediates. And let's see. So in this patient, because of patient does not have an allelase B, so the fructose 1-phosphate will accumulate, right? Will be accumulated in the patient, and this fructose 1-phosphate actually is very important intermediate. And what this intermediate does is to inhibit the glycogen breakdown, right? Inhibit some enzyme we will talk about later, the glycogen, the enzyme that break down glycogen. So if the patient consume a lot of fructose, and because the fructose cannot be metabolized, completely metabolized, right? Because of lack of the allelase, so will cause the accumulation of the fructose 1-phosphate, and this will inhibit glycogen breakdown and release glucose. So the patient usually have the problem with maintain blood glucose levels, because of you can, particularly under the fasting condition, right? You can break down glycogen to release glucose to raise blood glucose level. So they will have a problem with the hypoglycemia, and right. So you just understand the mechanism. Any questions so far? Right. Okay, so mitochondria. This is a very important organelle, right? And that's the oxidative metabolism, the energy plant, right? Called so, and also produce the ATPs like a TCA cycle, right? It occurs in the mitochondria, and the fatty acids oxidation. A lot of called a beta oxidation. We'll talk about later. It's also occur in the mitochondria. So a lot of oxidative metabolism, occur in the mitochondria. So mitochondria, if you learn already, right, in previous classes, so as a two membrane called inner and outer membrane, and inside of mitochondria is a mitochondrial matrix and cristae, and basically in the inner mitochondrial membrane. And in the mitochondria, the TCA cycle, the enzyme is located in the mitochondria, and also lots of enzymes and involve the respiration or phosphorylation, oxidative phosphorylation activity is also in the mitochondrial inner membrane. And pyruvate dehydrogenase complex, this you just need to know actually is an enzyme complex. I mean, this complex is composed of 3 different enzymes, right? And that also have some kind of prosthetic group that involved in the regulation of enzyme activities. And this enzyme complex, the main function of this enzyme complex is to dehydroxylate pyruvate and convert the pyruvate to acetyl-CoA, right? And this reaction occurs in the mitochondria, right? So pyruvate, generally from glycolysis. I just want to mention, pyruvate not just generated from glycolysis, but also generated from metabolism of amino acids, and and pyruvate enter the mitochondria. The first reaction will be converted to acetyl-CoA by the pyruvate dehydrogenase complex, right? And this enzyme, this reaction is very important, and also generate the 1-NADH, right, in the mitochondria. And this enzyme activity actually regulated by its self product. For example, glucose go to glycolysis and to pyruvate, right? And pyruvate being converted to acetyl-CoA and meantime produce an ADH. So if an ADH or acetyl-CoA levels increase, and this will inhibit the pyruvate dehydrogenase activity to allow the reaction to slow down, right? That actually makes sense, right? Acetyl-CoA basically can be completely oxidized for energy, and NADH is also represented the levels of energy, right? If you have a high levels of NADH, that indicate that your cell have a high energy. So cell always kind of design, to regulate, right? If you don't have enough, then you stimulate reaction to generate more. If you have lots of energy or whatever kind of product, then you want to slow down a reaction a little bit. So this is again the sort of a cell regulated mechanism. So again, acetyl-CoA and ADH are the product that can be can inhibit pyruvate dehydrogenase, right, to reduce the reaction. And the other thing I want to mention that again, we'll talk about many times. The acetyl-CoA can also be generated from fatty acids oxidation and also amino acids oxidation, right? So acetyl-CoA again is the key intermediates that connect all three macronutrient metabolism. Let me just finish this slide, and when we so an acetyl-CoA, like I mentioned, right? Converted from pyruvate, and acetyl-CoA again is a key. A molecule can be oxidized, can be generated from oxidation, glucose, amino acids, and fatty acids. And acetyl-CoA also is very important building block for fatty acid synthesis, ketone body synthesis, and also cholesterol synthesis, right? So cholesterol, fatty acids, and ketone body all synthesized from acetyl-CoA molecules, right? And because of all this kind of connection, so again, this is critical link between three macronutrient metabolism, right? Between fatty acids, glucose. For example, we know that glucose or sugar can be converted to fat, right? So this actually is a very critical link. Glucose metabolized to generate acetyl-CoA, right? And acetyl-CoA can be used for synthesis of fatty acids. So this kind of connection between sugar and fatty acids. All right, so let's just stop here today.